In this video, I'm going to explain the fundamentals of glutamate transporter function, and hopefully I will be able to demonstrate how cool these transporters are and how important they are in the brain. I will also describe a potential treatment strategy that I am researching in my laboratory that targets these glutamate transporters for neurological disorders. These disorders can include stroke, epilepsy, addiction, and neuropathic pain, amongst others. Glutamate is the main excitatory neurotransmitter in the central nervous system. By this, I mean that glutamate increases the chances that a neuron will fire an action potential. While glutamate is essential to cognition, learning and memory, and development, the levels of glutamate must be balanced with that of GABA, which is the main inhibitory signal in the brain. If this balance between glutamate and GABA is disturbed, and there is more glutamate in the synaptic cleft, excitotoxicity can occur, which is a condition that results in neuronal death. The levels of glutamate are mainly regulated in the brain by EAT2, or excitatory amino acid transporter 2. EAT2 transports glutamate out of the synapse and into astrocytes, where it can be converted to glutamine and other metabolites. EAT2 is a transmembrane transporter formed from 3 protomers, which consist of a transport domain and a scaffold domain. The transport domain slides up and down the scaffold domain to remove glutamate from the synaptic cleft. And it does this in a way that is referred to as an elevator motion. My lab has identified positive allosteric modulators of EAT2, or called PAMs. These PAMs increase EAT2's activity, which can lead to more glutamate being removed from the synaptic cleft, restoring the balance of glutamate in the CNS. Our lab has evaluated PAMs in preclinical in vitro and in vivo models, and we have seen that EAT2 PAMs are neuroprotective, can lessen neuropathic pain, and can increase drug acquisition. I am currently working on in vitro models of stroke and epilepsy. While we have demonstrated beneficial effects of EAT2 PAMs in preclinical models, currently we only have a general idea from medicinal chemistry and computational modeling of the allosteric binding site of PAMs on EAT2. We are now trying to further our knowledge of the specific biochemical interactions between our compounds and this transporter. To map the allosteric binding pocket of our PAMs, I am using amino acid mutagenesis and glutamate uptake assays. In this approach, I mutate one amino acid in the peptide sequence of EAT2, and then I see how the mutant transporter changes pharmacological variables such as potency and efficacy in the presence of our PAM. Based on how the pharmacological variables change in these assays and in collaboration with other approaches such as crystallography and SMFRET, we can map how PAMs are interacting with EAT2. Understanding these structural interactions can inform future development of EAT2 PAM therapeutics for many neurological disorders.